Today's lecture, we're going to cover part 2 of cell communication, and specifically we're going to talk about receptor tyrosine kinase pathways, talk a little bit about nitric oxide as a signal molecule and a specific pathway it's involved in, which is vasodilation of blood vessels, and also talk a little bit about hormones and specifically hormones binding to transcription factors as an intracellular receptor. Protein-Tyrosine phosphorylation as a mechanism for signal transduction appeared with the evolution of multicellular organisms. And essentially, when we think about protein-tyrosine phosphorylation, these are specific protein kinases that actually add an inorganic phosphate group to tyrosine residues on specific target proteins. In fact, they have identified approximately 90 different protein kinases that are encoded in the human genome, many of which are actually protein-tyrosine kinase receptors or are also cytoplasmic protein-tyrosine kinases. Protein-tyrosine kinases are involved in many different processes in multicellular organisms, such as regulation of cell growth and division, differentiation of cell types, survival of cells, and also migration. And like I stated just a second ago, there are receptor tyrosine kinases, and there are also cytoplasmic or what are called non-receptor protein tyrosine kinases. This particular diagram to the right is highlighting a receptor tyrosine kinase that's actually binding to growth factors in this particular scenario called PDGF. And the receptor itself is shown here. There's actually two of them, and these two receptors are dimerized. It means that they're interacting. And in fact, during dimerization, they actually led to phosphorylation of key tyrosine residues on each of those two receptors, and that led to docking of key proteins at those phosphorylated sites. So we're going to talk a little bit about mechanisms of activation of receptor tyrosine kinases, and then specifically talk about some types of proteins that are targeted to these receptors upon their autophosphorylation events. So the activation of receptor tyrosine kinases that are involved in signal pathways and cell communication, there's two different mechanisms that have been recently proposed, and we can talk about the first on the left. And specifically, one potential mechanism is that the signal molecule itself exists with two binding domains on it, and these two binding domains will interact with their binding sites on two different receptors. And when they do so, this will force two receptors to come together to actively what's called dimerize. This leads to ultimately what's called trans autophosphorylation. They'll actually cross-phosphorylate each other due to activation of their catalytic domains. And once these phosphorylation sites have been added, that will lead to docking of specific proteins, and those proteins will thus become activated and potentially activate other downstream targets. So some particular scientific research looking at these different types of receptor tyrosine kinases implicate this particular model, and others, however, implicate this other model here on the right, where a ligand could bind to two receptors. So we have ligand bind to one, ligand bind to a second, and what happens is when ligand binds to two separate receptors, as you're seeing here, it causes actually conformational state change on the surfaces in which these receptors will then interact. So it causes a conformational state change that now allows for a greater affinity for these two receptors to join together, leading to dimerization interaction, which leads to trans autophosphorylation, where we'll activate the kinase domains on those receptors, and then they will thus phosphorylate specific tyrosine residues on the opposite receptor, and this leading once again to docking of specific target proteins. So regardless of the initial dimerization, whether it be one ligand that binds to two receptors at the same time, or separate ligands that lead to a conformational state change that now lead to a higher affinity for these two receptors, regardless that activation occurs due to this dimerization, which leads to cross-phosphorylation, and that serves for docking sites of specific proteins. And the specific proteins that bind to these phosphorylated residues have been found to have proteins that essentially have important domains called SH2 domains or PTB domains. Now, proteins with the SH2 domain, or what's called phosphotyrosine binding domain, SH2 domain is also called the circumology 2 domain. These specific domains ultimately have specific binding sites for that the phosphorylated tyrosine with specific amino acid residues surrounding it. So they have a specific kind of binding peptide that these SH2 domains will bind to that include that phosphorylated tyrosine. Now, what types of proteins contain SH2 domains or phosphotyrosine binding domains? There are proteins that are considered what are called adapter proteins that will basically bind to that particular phosphotyrosine, but then also will help lead to binding to other proteins that leads to downstream activation of those proteins. There's also what are called docking proteins that will dock and essentially also get phosphorylated, which can then lead to binding of other proteins to those particular proteins. And then it could lead to binding of other kinases that contain those SH2 domains. So a kinase itself could have an SH2 domain, and binding to the receptor tyrosine kinase could activate that particular kinase. Or it could also lead to activation of transcription factors, such as a transcription factor actually having an SH2 domain or a PTB domain, and leading to its activation by interacting at a receptor. So these different figures here show you the different scenarios. Here's an example of a adapter protein being activated by a receptor tyrosine kinase. And you can see here that GRAB2 would be considered an adapter protein, and GRAB2 interacts with the tyrosine kinase receptor through its SH2 domain, which then leads to interaction with SOS, which is also called GAB, and it can then lead to activation of a very important G protein called RAS. An example of a docking protein activated by receptor tyrosine kinase is shown here in this particular scenario with a protein called IRS. And IRS happens to have a PTB binding domain, and once it docks at the receptor then can phosphorylate target sites on the IRS protein, and that then leads to binding and activation of other proteins at those phosphorylation sites on that docking protein IRS, and so forth. And here's an example of activation of a transcription factor such as STAT. STAT is a quite known transcription factor. When it binds to a receptor tyrosine kinase, it can get phosphorylated, which actually can lead to a dimer of STAT forming, and that dimer will then move into the nucleus and lead to altered gene expression. And this is also showing you activation of other types of kinases, which you can see here: PI3K, phospholipase C, and SHP2 happens to be a phosphatase. So there are different types of proteins that ultimately can be targeted to the receptor tyrosine kinase due to these phosphorylation events on these tyrosine residues. And obviously they're going to bind to by the specific receptors by binding at the tyrosine residues, as well as interacting with amino acids that are directly near or adjacent to that particular amino acid. So they do bind to specific peptides that contain that tyrosine, that phosphorylated tyrosine. Now here's an example of an SH2 domain shown in red binding to a specific peptide that happens to have a phosphorylated tyrosine residue. So you can kind of look at the interface between this SH2 domain and this peptide, which is shown in yellow and green. And the sequence specifically this is SH2 domain is interacting with is a proline asparagine, tyrosine, glutamate, proline sequence. So ultimately, the phosphorylated tyrosine is actually shown a little bit lower here. It binds into a certain pocket of the SH2 domain, but you can see that it also, part of the SH2 domain binding to this particular sequence, also makes important contacts with other amino acids as well that are within a few amino acids of the sequence of that phosphotyrosine. Either they're right adjacent to it or a couple amino acids down from it. Ultimately, this is going to lead to that high affinity binding of that SH2 domain to the a-specific sequence on the receptor. So it really highlights the interaction between these an example, an SH2 domain with a specific peptide that contains that phosphotyrosine. So different SH2 domains have specific peptides in which they bind to allow for specificity as to which particular proteins with SH2 domains get activated by certain regions of the receptor of a tyrosine kinase receptor. So many proteins that have SH2 domains that are involved in binding to receptor tyrosine kinases also have what are called SH3 domains, which you can see here. This one happens to have one on the N terminus and also one on the C terminus as far as the SH3 domains. This happens to be GRAB2. And GRAB2, if you remember, ultimately is important for activating downstream RAS by SOS. So we're going to look at this a little bit later, but ultimately these SH3 domains are important for interaction of something like GRAB with SOS and then ultimately allowing for activation of some downstream targets. So the SH3 domains are for interaction with other proteins, and the SH2 domains are important for interaction with the receptor tyrosine kinase. Now, receptor tyrosine kinases, a very known pathway in which is stimulated upon activation of these different receptors is what's called the MAP kinase pathway. And ultimately, the MAP kinase pathway is very important in stimulating cell growth, cell division, and cell differentiation. And essentially, when we think about activation of these receptor tyrosine kinases by mitogens or growth factors, this ultimately can lead to activation once again of that GRAB protein that has an SH2 domain that binds to the receptor tyrosine kinase. And this then activates with or interacts with SOS, and SOS serves as a RAS activating protein, and SOS and GRAB will ultimately become activated and move to the membrane and activate RAS. RAS is a monomeric GTPase, so it's a monomeric G protein that binds to GTP and can hydrolyze it. In its GTP bound state, it's turned on. When it hydrolyzes GTP to GDP, it is turned off. So RAS is a G protein, just like the G protein linked receptor pathway we talked about in last lecture, except that the G protein linked receptor pathway had a trimeric G protein that interacted with that specific receptor. In this case, this is a monomeric G protein. RAS, what it will activate is what's called the MAP kinase pathway. And the MAP kinase pathway consists of three really important kinases. RAS will actually activate or help lead to activation of MAP kinase. And MAP kinase, once it's activated, will phosphorylate MAP kinase. And then MAP kinase will then phosphorylate MAP kinase, and this leads to phosphorylation of many different targets, such as gene regulatory proteins and other proteins such as enzymes, etc. So ultimately, there are many different targets that have been identified downstream of this particular MAP kinase pathway. And like I said, the MAP kinase pathway, one of the most important functions is that it's important for controlling cell growth. So ultimately, if a cell is going to be stimulated to divide due to interaction with growth factors or mitogens will interact with receptor tyrosine kinases and ultimately lead to activation of the MAP kinase pathway. Now, MAP kinase was originally identified and named RAF, And MAP kinase was originally named MEK, or MAP ERK kinase, and the MAP kinase was originally called ERK, and which was extracellular signal regulated kinase. Once these kinases were then identified as all being in the same pathway, they were ultimately renamed to MAP kinase, MAP kinase, and MAP kinase to simplify this particular transduction pathway. Now, MAP kinase is a serine-threonine kinase, so that means it phosphorylates actually serine or threonine sites, and actually MAP kinase is both a serine-threonine and a tyrosine kinase, which is why you see two different sites being phosphorylated on MAP kinase. So ultimately, when we think about kinases, they can either phosphorylate tyrosine residues or usually serine threonine. In this case, you saw that MAP kinase actually can phosphorylate both types of amino acids. So what helps control activation of RAS? So remember I told you RAS will be activated by GRAB and SOS. Now RAS, to turn on, binds to GTP. So in its inactive state, it's bound to GDP. And it has to exchange GDP for GTP to become active again. And there are important guanine nucleotide exchange factors that essentially help speed up the rate in which RAS exchanges GDP for GTP and becomes turned on. This is similar to what we saw with SARF1 when we talked about COP2 vesicle protein coat assembly, that there was a guanine nucleotide exchange factor that helped allow for SARF1 to exchange GDP for GTP. Guanine nucleotide exchange factors are also involved in helping increase the rate in which RAS becomes active. SOS is a guanine nucleotide exchange factor that is ultimately going to help activate RAS. So SOS is an example of a guanine nucleotide exchange factor. There's also other types of proteins that can interact and regulate the active or inactive states of RAS as well. There are what are called GTPase activating proteins, and GTPase activating proteins, also called GAP, are proteins that actually kind of help speed up the rate of hydrolysis of GTP by RAS. And it's often called proteins that actually kind of put a timer on something like RAS so that ultimately it's turned off after a certain period of time. So GAPs, or GTPase activating proteins, shorten the actual time in which it takes for RAS to actually hydrolyze GTP to GDP. So essentially it's going to speed up hydrolysis of GTP by RAS and essentially turn it off. There are also what are called guanine nucleotide dissociation inhibitors, and if these proteins interact with RAS, then they actually inhibit RAS's ability to exchange GDP for GTP, which essentially inhibits the activity of RAS. So ultimately, the presence or absence of these important activating or inhibitory proteins are really going to help regulate the activity of RAS in cells. Why is RAS important? RAS has been found to be mutated in what we call a constitutively active form or overactive form in about 30% of all cancers. So RAS is considered what's called a potential oncogene. And a potential oncogene in cancer development is basically a gene that stimulates the cell cycle, or a gene that stimulates the cell to divide. And when it becomes an oncogene, it means that that gene has acquired a mutational change that ultimately make the protein product of that gene overactive, always on, and essentially that can lead to potential formation of cancer. So ultimately, RAS is a critical protein for stimulating cell division, and its conversion to an oncogene could potentially have drastic effects on control of the cell cycle in cells with that converted oncogene in something like RAS. Now, as an example of a known receptor tyrosine kinase the insulin receptor is a receptor tyrosine kinase. So this is a good example of a receptor tyrosine kinase. It's a little different structurally than what we just viewed a little bit ago. And you can see that the receptors themselves are actually already interacting together, which is something we didn't see from the previous model looking at receptor tyrosine kinases. So they are a receptor tyrosine kinase, but the interactions of the receptor are a little bit different than the two different mechanisms we previously discussed. The receptors themselves, the two receptors, consist of two domains, an alpha domain and a beta domain, which you can see from the picture. And these two receptors are actually bound together through their alpha domains by disulfide bonds. The alpha domain of each of the two receptors is actually on the extracellular side of the membrane, and the beta domains, are actually embedded in the membrane, and there's a portion on the extracellular side and on the cytoplasmic side, which you can see. So ultimately, these two receptors are interacting together in the absence of something like insulin. However, when insulin binds to them, it causes dimerization of the receptors due to a conformational state change. So they'll actually move closer, and then you'll get phosphorylation of the receptors, which ultimately means that you're going to get cross-phosphorylation due to activation of those kinase domains in the receptor, and that will lead to docking of particular target proteins. Now, insulin, as a signal molecule, it's released when we have higher blood glucose levels. And beta cells in the pancreas will ultimately release insulin upon increased levels of glucose in the blood. And ultimately, the ultimate response of that is insulin will then be released into the bloodstream and will help serve as a signal molecule to tell cells to uptake glucose into the cells to ultimately help reduce the glucose levels in the blood. So insulin here will bind when there are higher glucose levels, then insulin will be released by the pancreas, the beta cells in the pancreas, and then insulin will travel around, and if there are insulin binding receptors on certain cell types, then ultimately insulin will bind to this receptor tyrosine kinase and lead to activation of the receptor, the dimerization of these two receptors. Now, when we think about activation of downstream targets, one known downstream target of the activation of this receptor tyrosine kinase by insulin is a protein called IRS. And IRS is shown here in green. And IRS has a binding domain that's a PTB, the phosphatyrosine binding domain. And this is the insulin receptor substrate is what IRS stands for. So insulin receptor substrate. It binds to the activated insulin receptor, this receptor tyrosine kinase, through its PTB domain. And you can also see that it's a docking protein that's going to also have key tyrosine residues that ultimately also get phosphorylated when it docks onto this receptor. So the receptor itself is going to ultimately phosphorylate specific sites on this docking protein, which will then lead to binding of other proteins. And oftentimes the other proteins that bind to these phosphorylated sites on IRS are proteins that have SH2 domains, such as GRAB, which we saw before, that is activated on receptor tyrosine kinase receptors, and then ultimately that could lead to activation something like RAS. So you can see different types of proteins that are ultimately activated by phosphorylation of specific sites on IRS that's docking on the insulin receptor. One of the important impacts of phosphorylation of IRS is ultimately it can lead to activation of a kinase called PI3K. And PI3K is an important kinase because it's important in glucose uptake and also stimulating glucagon synthesis. When we look at IRS and ultimately binding, its ability to bind to the insulin receptor tyrosine kinase. You can see its important phosphotyrosine binding domain that's going to be critical for binding to the receptor. And you can also see several different tyrosine residues that are found throughout the middle portion of the protein as well as towards the C-terminus of the protein. And you can see specific phosphorylation sites of these tyrosine residues that are important for binding of specific target proteins, such as PI3, GRAB2, and the SH2, which is a phosphatase. So when we think about the role of this particular receptor, the insulin receptor, in helping bring in glucose, we can kind of look at what's known about the pathway. So this is showing you the insulin receptor in pink, and the insulin receptor, when it binds to insulin due to higher blood glucose levels, that leads to activation of the IRS, so binding of IRS and then phosphorylation of IRS, and that will then lead to activation of the target protein PI3K, and thus will then activate PDKI and PKB. And why is this important? Because this particular signal pathway has been found to lead to exocytosis of what are called GLUT4 receptors. Why is this important to exocytose these GLUT4 receptors? Because these GLUT4 receptors or transport proteins are ultimately involved in bringing glucose in. So if we have higher levels of glucose in the blood, what will ultimately be stimulated by the insulin receptor is exocytosis of these channels onto the membrane surface, and these channels ultimately will allow for movement of glucose into the cell. So ultimately allow for transport of glucose, so glucose can be brought in or by uptake into the cell to help reduce blood glucose levels, so ultimately will help supply glucose to the cells themselves. Now, when we also think about kind of defects in these in the insulin pathway, we can talk about type 1 and type 2 diabetes. Type 1 diabetes is ultimately due to defects in this pathway. And when we think about type 2 diabetes develops due to insensitivity to this to insulin by this pathway. So they're very kind of different as far as ultimately how they've evolved as far as defects. But ultimately, when we think about type 1, this pathway is defective, and ultimately in type 2, this pathway becomes desensitized or insensitized to ultimately insulin release. So what other signal molecules and pathways can we talk about? We can talk about so many, but we can restrict it to just a couple more in this lecture. Another important signal molecule is nitric oxide, which is a gas. And nitric oxide is both an extracellular and intracellular messenger. It has a variety of functions. It's involved in anticoagulation, neurotransmission, smooth muscle relaxation, visual perception. So it's an extremely important signal molecule, and ultimately it's produced in signaling cells by enzymes called the nitric oxide synthase. And nitric oxide, one important pathway or mechanism that it's involved in is ultimately smooth muscle relaxation, which is critical for vasodilation of blood vessels. Why is this important? Because ultimately, if we have high blood pressure in our blood vessels, NO will be produced and ultimately stimulate vasodilation or relaxation of smooth muscle, which will allow for increased blood flow through those blood vessels, which will help reduce blood pressure. Now, this is showing you that exact model. So when we think about blood vessels and essentially their anatomy, envision the fact that the blood vessels are lined by endothelial cells, which are shown here, and surrounding the endothelial cells are smooth muscle cells. Now, if the blood pressure becomes higher in our blood vessels, our nervous system will sense that change in blood pressure, and intervening nerves of the blood vessels will then release acetylcholine. Acetylcholine, when it's released by the intervening nerves, will interact with receptors on the endothelial cells, and this will stimulate production of nitric oxide. So ultimately, those endothelial cells become the signal molecule, or the cell that's actually releasing the signal molecule. And endothelial cells will make nitric oxide from L-arginine, and ultimately the enzyme that's important for that are the nitric oxide synthases. So nitric oxide will rapidly diffuse out of the endothelial cells into the surrounding smooth muscle cells, and that will ultimately lead to rapid relaxation of the smooth muscle, which leads to opening of the diameter of the blood vessels and decreasing blood pressure because there's an increased area for blood to flow. So to kind of give you even more details in relationship to the NO pathway, NO, when it's produced by endothelial cells, will move into smooth muscle cells, and its receptor that it will actually bind to is an enzyme called guanylyl cyclase. And when guanylyl cyclase binds to nitric oxide, it will lead to increased levels of cyclic GMP. And increased levels of cyclic GMP decreases actually cytosolic calcium, which leads to smooth muscle relaxation. So you can see in this particular scenario here's those endothelial cells producing NO. NO moves in, it binds to guanylyl cyclase, and it produces higher levels of cyclic GMP, which will lead to relaxation of the smooth muscle cells. NO also plays a role in male arousal, so erections, and ultimately NO is going to be released in that area that leads to vasodilation in that particular region or organ of the body. So besides the nitric oxide pathway and its particular role in vasodilation and help regulating blood pressure, we also have other types of signal molecules that activate other types of pathways. And one such signal molecule or one category are hormones. When we think about something like steroid hormones or thyroid hormones, one of their major receptors in which they bind are actual transcription factors. Here's some different examples of steroid hormones, thyroid hormones. Remember, steroid hormones, thyroid hormones in this particular structure are relatively hydrophobic, so they'll readily diffuse into cells. And one of their biggest targets are that they'll bind to intercellular receptors that actually are transcription factors. So this is showing you an example of a hormone, cortisol, binding to its intracellular receptor. And this intracellular receptor is a transcription factor. It will activate the transcription factor. That transcription factor will then bind to specific regulatory sequences and help increase transcription rate of key genes, which you can see in this particular diagram here. So these are relatively short pathways as far as receptor actually serving as a transcription factor and binding to key genes, turning on gene expression. So another example just describing this exact scenario is looking at something like the glucocorticoid hormone. And ultimately, these transcription factors that they bind internally as an intracellular receptor are normally inhibited, which you can see in this particular diagram. It's inhibited by HSP90. Upon binding of that intracellular receptor with the glucocorticoid hormone, it leads to a conformational state change that will then lead to release of that inhibiting molecule, protein, and it will now be bound to the hormone itself and will thus activate the transcription factor. You can also see that same inhibition and activation process as far as its change in conformational states also shown here on the left. An example of genes that might be turned on by a specific hormone, you can see estrogen being the hormone, the oviduct being the tissue site where the signal molecule is going to ultimately turn on a gene like albumin, and this is something that they've seen in chicken. So ultimately, this is kind of showing you a steroid, a tissue that has particular receptor for that steroid, and ultimately a gene that's turned on. So when we think about activation of signal pathways, keep in mind, remember, we're going to have potentially multiple signal molecules inundating cells at a single moment. And essentially, we can talk about the fact that signal pathways can have convergence, divergence, and crosstalk. When we think about different signal pathways, remember that we can have different signal molecules binding to different types of receptors, which you can see here. And they can activate different pathways, but there can also be crosstalk between them. So different signal molecules that can actually interact with both pathways. They can lead to activation of different continuous pathways, such as divergence. And they can converge and activate the same signal molecules. So we're going to kind of look at that in a little bit more detail. So when we think about something like crosstalk essentially, some signal molecules may influence each other's pathways. So here's showing you one receptor, here's another receptor binding to different signal molecules. And something like activation of pKa by cyclic AMP in this particular pathway could ultimately lead to activation of the same protein that also could be activated by this particular pathway. So we're seeing crosstalk between pathways. Ultimately, different signal molecules can influence each other's pathways. Convergence, an example is something that we see with convergence of pathways. We can see that multiple receptors could potentially activate the same type of protein, such as activation of something like GRAB2. So we saw GRAB2 and its interaction with receptor tyrosine kinases, but GRAB2 have also been shown to be activated with G protein coupled receptors and also something like integrin receptors that are involved in binding to extracellular matrix. So ultimately, if you activate the same type of downstream target, then potentially that could then activate another downstream target such as RAS. So GRAB2 could be activated by all three different receptors in this case, which ultimately leads to activation of RAS or something like the MAP Kinase Pathway. And divergence, an example of divergence is ultimately when we think about we have these different receptor pathways that have different target proteins that they activate in different end responses for these different signal pathways. So keep in mind, this is trying to ultimately describe the higher complexity of signal pathways, that they aren't just simply a single pathway and activating just this target, that ultimately these different pathways oftentimes can converge, they can diverge, or there can be crosstalk between them. So cell communication pathways become extremely complex, which is, if you think back at that figure I showed you in the cell communication lecture, with all the different signal pathways and all the different cross talk potential points, you can see how complex cell communication can truly be.